Hello everyone. Today we will discuss about the support media that is very important in gel electrophoresis. Basically, this support media provide the matrix in which the separation of the macromolecules takes place. If there is no supporting media, then molecules might would freely move through the buffer, leading to no separation or poor separation, or there might be a less resolution of the sample. So to avoid these things, like to avoid the low separation or to avoid the unwanted diffusion, we need the support media so that we can get a better result. So today in this video, we will discuss about the support media in gel electrophoresis. But before moving forward, if you are new here, please like and subscribe to this channel so that you cannot miss any future video. For the very first time, the electrophoresis was performed on the protein samples, and this technique was first utilized or first used by the Arne Tiselius, and for that he got a Nobel Prize in the year 1948 in chemistry. Basically, there were no any support medium. However, it was soon realized that many of the problem that was arising at that time for the separation or for the visualization of the sample was due to the or was because of the diffusion of the sample in any direction, and the another problem is the convection current, because since the protein is freely kept in a free solution, so there is a chance that these protein molecules can move in any direction. So to avoid or to minimize this problem, many scientists has utilized or used the medium that is called as the stabilizing medium or stabilized medium. And these medium looks like this, a porous membranous medium called as porous mechanical support, which was wetted in a electrophoresis buffer and in which the electrophoresis of the buffer ions and the sample could occur like this. So this is the porous medium support kept in a electrophoretic buffer sample, and these are the wells where we can pour or we can fill our sample so that the samples can run in particular direction, and by utilizing this type of supporting media we can avoid the diffusion of the samples in any other direction, and also we can minimize the chances of convection current. This type of support media cut down the convection current and diffusion so that the separated compound remain in a sharp zone. The earliest support used were the filter paper or the cellulose acetate strip, or you can also use the silica or alumina at that time. And these support mediums were wetted in a electrophoresis buffer. Nowadays, these mediums are infrequently used, although in particularly for many years, small molecules such as amino acids or peptides or the carbohydrates were routinely separated and analyzed by electrophoresis on support mediums such as the paper um, or thin layer electrophoresis cellulose plate or the silica or aluminum plate. While the use of filter paper or thin layer supports are fine for resolving the small molecules like small peptides, but the separation of the proteins, especially the macromolecules, like the larger molecules, that is the proteins and the nucleic acids, on such support is not very good. They such type of support system gives us a poor result. So after the introduction of the use of gel as a support medium, we got a rapid improvement in methods for analyzing the macromolecules. Which system? The gel system. Gel system. Basically, in earliest days or in early days, the gel system used was the starch gel system. And still we are using this gel system, but there are many drawbacks, or there are some limitations for this type of gel system. That is, first of all, it is a very time consuming and labor intensive gel system. Gel system requires a large sample size, and also there is a limited protein variability, and these are costly also. Therefore, this system is a complex system. So nowadays we are using a Agarose gel and amide gel. Amide gel. So these are the two gel system which we are using these days, and these are cheap, cost effective, and we can prepare our gel in very less time. So let us start with the Agarose gel system. So as the name suggests, agarose gel are formed by suspending a dry agarose in aqueous solution. So what we need to add agarose where in buffer aqueous solution, right? This buffer can be TAE or TBE. We will discuss about these buffers later on. So suppose we have a agarose powder. So this bottle contains the agarose powder. So from this bottle, we weigh particular amount or the required amount of the agarose, and then we put those agarose powder in a flask, and then after that, in that particular flask, we will add TA or TBE buffer. Then after adding the buffer, since agarose is insoluble at room temperature, so to make it solubilize, we need to boil the agarose with the buffer at a particular temperature. And after that, we get until like we get a clear solution like this. So as soon as we get a clear solution like this, we will wait for around 5 to 10 minutes so that we can get a normal temperature for this solution, because in this solution we need to add ETBR. We will also discuss this ETBR later on. And after adding the ETBR into this solution, then we are supposed to pour this particular solution into a casting tray. That we will wait for around 30 minutes at a room temperature to obtain a gross gel. Now question arises, what actually the agarose is. So agarose is a linear polysaccharide, and it is having a average relative molecular mass of about 12,000. And this agarose is made up of, or it is a basically a repeating unit of agrobios. What agrobios is? Agrobios comprises of alternating units of galactose and 3,6 and hydrogalactose. This is the structure representing the galactose and 3,6 and hydrogalactose. This is the structural formula of agarose. So what agarose is? Agarose is a basic repeat repeating unit of agrobios. And what agrobios is? Agrobios is comprising of alternating unit of galactose and 3,6 and hydrogalactose. If we talk about only this much, so this is the monomer form of agarose. Monomer agarose. And if we consider this N, so this N is the polymerization unit that represents the repeating form of this monomer agarose. So if we consider this N whole, then the whole term is the polymer form of agarose. Agarose. All right. So this is the structure formula of agarose. Agarose is one of the component of the agar, and this agar we can obtain from seaweeds. So it is a mixture of the polysaccharides. Agar is a mixture of the polysaccharide, and agarose is one of the component of the agar, and we can obtain this agar from seaweeds. These days we can utilize agarose in a composition or in a concentration of about 1 to 3%. 1% means 1 gram of agarose in 100 ml of TAE buffer or TBE buffer. All right, 1 to 3% means we can add 1 gram of agarose, or we can add, like, if we are talking about 2%, that means we can add 2% of, we can add 2% or 2 gram of, like 2 gram of agarose in 100 ml of TAE or TBE buffer. And after adding this amount of agarose in TB or TA buffer, then we are supposed to boil those mixture, and then we will pour it into the casting tray. Let's suppose we are let's suppose we intend to make 1%. Then for the 1%, we will be adding 1 gram of agarose. For 1%, we will be adding 1 gram of agarose in 100 ml of TAE buffer. All right, like as it is for 3%, if we talk about. So for the 3%, we will weight 3 gram of agarose, and then we will add those 3 gram of agarose in 100 ml of TAE or TBE buffer. So like this, we can obtain our agarose gel. This gel-like property of agarose gel is just because of the presence of the inter and intramolecular hydrogen bonding. Inter and intramolecular. So this is the hydrogen bond present. Inter, that is the bonding which is between the two different molecules, like one this and another one is this one. So this is just because of the presence of the both inter and intra. This is the intra, because the intra means it is within the molecule, and this one represents the intermolecule, and this represents the intramolecular hydrogen bonding. So the gelling property of agarose gel is because of the inter and intramolecular hydrogen bonding within this one, within and between the long agarose chain. So this is the long agarose chain. So this is the one molecule of agarose, another molecule of agarose, and it goes on like this. When these two chains are joined together by hydrogen bonds, by the interhydrogen bonding, then it creates a mesh-like structure. So this represents the monomer of agarose. But if we dissolve it in a buffer and then leave it for cooling, then it creates a mesh-like structure like this. And if we leave the buffer, leave the solution for a longer period of time in a casting tray for like about 30 minutes or more, then further it creates a more compact and condensed structure like this. So this is the mesh of agarose gel, and this mesh like structure creates pore in between. So these are the pores, and this cross-linked structure gives the gel good anti-conventional property. Anti-conventional properties means these cross-linking gives a hydrogel. These are the hydrogel, actually a 3D network structure. This is the 3D network structure, and making them insoluble. These are insoluble unless or until you heat it at particular temperature, that you give it a boiling temperature, right? So until and unless you give it a boiling temperature or a heating temperature, if you leave the gel, if you leave this mesh or 3D like structure at a room temperature, it would be insoluble, and hence these insoluble cross-linked structure allows the effective immobilization, and also it helps in the release of the biomolecules, because you want that your biomolecules travel across it. So at the end, what you want, you want to retract your biomolecules from this mesh like structure for further studies. So this type of structure allow the release of the biomolecules. But if you want to again remelt and you want to get the monomeric form of this structure, then when you heat it at at around like 100 degrees centigrade, then again you will get the monomeric form of this agarose. The pore size in agarose gel is very crucial for the macromolecule separation, as it dedicates the ability of the DNA fragments to pass through the gel matrix. Like if the smaller pores, then the smaller DNA molecules or the smaller RNA or smaller protein will be passing through it, and then only we will be getting a better resolution. And if the larger pore size, that means it facilitates the passage of the larger DNA molecules. So the pore size in the gel is controlled by the initial concentration of the agarose. If the concentration of the agarose, if the concentration of agarose is high, then we will get a smaller pore size. And if the concentration of agarose is low, then we will get larger pore size. Alright? As it is saying here, the larger pore size are formed from the low concentration. Means, for example, if we are making 1% agarose gel, means we will be adding 1 gram of agarose in 100 ml TAE buffer. And another gel we are making of 3% agarose gel. Alright. So for this we will be taking 3 gram of agarose in 100 ml of TAE. So here the concentration of the agarose is less, here the concentration of the agarose is more. Alright, here the concentration is less, here the concentration is more. So when we use this concentration, we will get bigger pore size in comparison to if we are using 3% agarose gel. So in this condition, we will get bigger pore size. Here the pore size will be smaller. Like as you can see here, this is the larger, then this is the medium one, and this is the smallest one. So for this, let's see, for example, for the 1%, so this is the 1%, this is maybe for the 2%, and this one is maybe for the 3%. All right. So likewise, suppose this is the agarose gel, and this is of having of percent 1 or 2%. Suppose this is a 2% agarose gel, and we want to separate our DNA or RNA molecule. So RNA are basically suitable for separation by using the 2% agarose gel. So if we want to isolate our, or we want to separate our RNA molecule, then the RNA molecule, suppose this is the size of the RNA molecule, and another molecule that is of size this one. So this will move easily across this type of mesh, not this one. This will stuck somewhere here only. This will not move or not pass across these gels. All right. So the size of the pore is very crucial, because the agarose gel separates the DNA fragment based on their size, with smaller fragments moving faster through the gel because it have a suitable pore size and migrating faster than the larger fragments. All right. Let's say we have different um, macromolecules in respect to size. Suppose these are the different macromolecules in which we are interested. So these are the macromolecules. All right. And we want to separate these macromolecules. So to separate these molecules, what we need to run these sample on this agarose gel. So if we run this macromolecule on this type of gel, then the smaller one will move faster, because the pore size is very much suitable for this type of or this size of macromolecules. So the smaller will move faster and will be ahead of the this medium one and this the larger one. So the smaller, then the medium, and the bigger one. Let's say we are interested in resolving small DNA molecules, maybe of size 200 to 50 base pair. So for this type of DNA or for this size of DNA molecules, high agarose percentage is required. So high agarose percentage is required. Example, 2% is suitable for resolving the smaller DNA molecule of size 200 to 50 base pair. Pore size will be the smaller in this condition. And also we are interested in resolving comparatively larger DNA molecule, maybe of size 0.2 to 10 kb. So to resolve this size of DNA molecules, we will need 0.7 to 1% agarose concentration. And hence the size of the pore will be bigger in comparison to the 2% agarose gel. Here the size will be comparatively bigger. And also if we want to resolve very large DNA molecule, like in comparison to the previous one, so for example of maybe the greater than 10 kb. So for this size, we need much greater pore size, maybe of percentage 0.1%. But these type of gels are very fragile and will be very difficult to handle. Lastly, the purity of agarose is also very important for better separation and resolution of DNA or RNA or protein molecules. So the purity of the DNA molecules totally depends upon the type or the sugar residues that are present with the agarose molecules. Like although the essentially like the agarose is available in a free form, that is this, but the substitution of the alternating sugar residues like the carboxyl, methioxyl, pyruvate and sulfate group plays a very important role. Because of the presence of these groups, the substitution can result in the electroendosmosis during the electrophoresis. So what is the electroendosmosis? It is a very important phenomena because it allows the flow of water and its content through the porous material like gel under the influence of the electric current, and this is caused by the migration of the ions associated with the fixed charged groups on the matrix. So if these molecules will be or these residues will be present on the agarose gel, then definitely this will hinder the electroendosmosis phenomena during the electrophoresis, and hence the ionic interaction between the gel and the sample. So we does not want to face this type of effect, but in other situations like to study or to do particular type of research, then it's fine to use these type of agarose gel that carries such residues along with it. So agarose is therefore sold in different purity grade from high to low purity grades, and this purity grade totally depends upon the presence of these residues, especially the most commonly residues which are present in agarose these days is the sulfate group. All right. So the lower the sulfate content, the higher the purity. Lower the sulfate, higher will be the higher purity for the agarose. This is all about the agarose gel. And in the next session, we will discuss about the polyacrylamide gel.